Hello students, this is Dr. Faizan Mirza. I'll be discussing with you in this video the questions related to mammalian transport. I'll be covering the details of questions that show you photomicrographs of blood cells, and you are to identify the red blood cell, white blood cell, the lymphocyte, the neutrophils and monocytes, and the arteries and veins and what so. So let's move on to this question. This is May June 2019. This is from May June 23. This is question number 5. So this photomicrograph is given to you. They are saying there's a photomicrograph of a human blood cell. They have been placed in a solution of sodium chloride. So if you look closely, you can see that the red blood cells have crenated. These spikes can be seen. Some of the red blood cells are very much intact. Some of them have become crenated. Some of them become very crenated. So you can see that if they have become crenated, that means that water have gone out, water left these cells, and what if water left these cells, it means that there was a high solute concentration outside in this solution. So that was a solution that was having hypertonic sodium chloride solution that probably you can just mention here. If we come to the cells here, so if it's having a bean-shaped nucleus, if it's a large kidney-shaped nucleus, a large cell, that's a monocyte. If it's having a lobe nucleus, so that means be a neutrophil. If the cell is having a nucleus which is very large and it fills up almost all the cell, so that's a lymphocyte. So you can just mention all these things accordingly in this diagram, according to the figures which is mentioned. State the function of the cell labeled by its function is to carry out phagocytosis because it's a neutrophil. The red blood cell then figure as described is created. It has become abnormal or shriveled in appearance. Then how the red cell became created. So the solute concentration outside is the cell is higher, is compared to inside. The red blood cell bottom moves out red blood cell into the solution by osmosis down the bottom initial gradient. This will cause the red blood cell to shrink and shrivel. Researchers studied the effect of altitude on the red blood cells of humans, and they are saying recorded the healthy and the population of them living of red blood cell at different altitudes. Population A, B, C, D are mentioned. The mean red blood cell count is given. Altitude is given to you as well. Calculate the percentage increase in population A and population D. So population A is 4.5 and population D is 5.1. We just calculate percentage increase. So this is 5.1 minus 4.5. We'll calculate the change, divide by 4.5, the actual value or the initial value. So we'll just multiply by that 100 and we'll get the answer. We'll mention the answer here in percentage. Describing this and suggest the explanations for the data shown in table 5.1. We know at that sea level there is a high PO2 available for hemoglobin to get fully saturated. At high altitude, the PO2 is partial pressure of a gas. Partial pressure is the measure of a quantity of a gas in a particular environment. So at high altitude, the PO2 in the atmosphere is very low. In order to achieve complete hemoglobin saturation, RBC count need to increase. Bone marrow is stimulated to produce more red blood cells because we know that inside the bone marrow they are stem cells, so they divide by mitosis. This adaptation assures that better oxygen availability to the tissues at high altitudes continue. Carbonic anhydrase enzyme is found in the red blood cell, which has an important role in transporting respiratory gases. We know the function. You can just recall the fact of what carbonic anhydrase was doing in your cells. So explain why the non-competitive inhibitor of carbonic anhydrase would reduce the supply of oxygen actively to the actively respiring tissue. The binding of the inhibitor would reduce the Bohrs effect. As carbonic anhydrase will stay inhibited, chances of HHB formation will be low. We know when hemoglobinic acid forms, the hemoglobin loses a lot of oxygen. So if hemoglobinic acid persists, if hemoglobinic acid formation will be low, it means that oxygen delivery will be low as well because carbonic anhydrase enzyme stays inhibited. As a result, the oxygen moving to the respiratory tissue will only be down the gradient, and the Bohr's effect will be overall reduced. Let's move to the next one. This was a question of variant 20, variant 23. You can see artery and vein, both are shown to you. You can see the arteries having 3 layers and veins having 3 layers as well. So artery is always having a wavy lining in the inside that allows stretching, and that allows when the artery gets stretched because of the blood filling in, it doesn't break away. The inner lining doesn't break away because of the shear force which is generated because of the artery being stretched. So these are the 3 layers of the arteries, whereas vein will also have the 3 layers. So vein, you can see that this vessel Y is a vein because it is having a larger lumen. So yes, you can just add all the labels here. So just for your own education, so we know X which one is artery, which one is vein. They're asking you straight reason for identification of the type of blood vessel shown by the figure Y. This tunica media is thin. There is no wavy lining in the internal elastic lamina. Lumen is irregular, large collapse, thin walled vessel, large size vessels. That must be vein. They are saying tissue fluid and lymph are formed by when the blood arrives in the capillary network of the tissue. Actually, white tissue fluid is more similar to blood plasma than blood. So it is blood. The blood is having all the cells, red blood cell, white blood cell, platelets, as well as plasma proteins. In tissue fluid, red blood cell, large proteins are absent. Example, albumin. White blood cell antibodies are small solutes, are dissolved in plasma as well as in the tissue fluid. So you just need to compare tissue fluid, and you need to outline how it is more similar to blood plasma and not to blood, because everything that's in blood plasma leaks out into the tissue space to form tissue fluid, except for the large proteins. As soon as the tissue fluid formed in the blood arrives at the capillary network at a higher concentration of amino acids rather than the forming the lymph draining away from this network. So yes, when the capillary delivers the amino acid will be small and they will pass out in the tissue space. The tissue cells will start to use the amino acid to build new proteins. And when these have been used up, the blood leaving from the capillary network will have low amino acid concentration. Further, they are saying trachea and bronchi, bronchioles in this gas exchange system, they require supply of glucose and oxygen from the blood supply for the function of smooth muscle. You need to recall what smooth muscles are doing, outline the function of smooth muscles in the gas exchange system. So smooth muscles, they contract. And when they contract, they allow the diameter of bronchioles to be maintained. So they allow bronchodilation, bronchoconstriction. Smooth muscles also provide support to the gas exchange system, trachea, bronchi, bronchioles. So yes, that's the function of smooth muscle. They can stay contracted for a long period of time. Here, this question from May, June 2018, Variant 21. They have shown to you the ribbon model of hemoglobin molecule. They're asking you to label what each of these represents. So E is a chemical group that is not made of amino acid, that must be a prosthetic group. F of they are saying a form of secondary structure found in polypeptide hemoglobin. So this F, you can see this is coiled, so this must be alpha helix. Tertiary structure, why we're saying G is tertiary structure? Because each polypeptide is folded to form complex 3D shape. So each polypeptide in 3D shape is a tertiary structure. We are using they're asking about H. So each polypeptide is made about a sequence of amino acid. Whenever this is asking about sequence or order of amino acid, it's always primary structure. Each hemoglobin molecule is composed of 4 polypeptides, shown here by different shading. So if 4 multiple polypeptides join together, that must be quaternary structure. So you just mention each for these, and you get 5 marks here. The effect of carbon dioxide concentration on percent saturation of hemoglobin oxygen was investigated. A sample of mammalian blood was exposed to gas of mixture that was screened for increasing partial pressure. So this information already we already know this. So this is the oxygen dissociation curve. You can see PO2 is increasing, and here the oxygen saturation is increasing. This graph, which is towards the lower side, it is at a higher PCO2, which is 10.7 kilopascal, whereas the graph, which is at the above side, or you can see this graph, this is at a lower PCO2, which is 5.3 kilopascal. So they are saying we know the if the tissue is having a high PCO2, it will end up receiving more oxygen as compared to a tissue which is having low CO2. So if I say this is tissue X and this is tissue A, so tissue A will receive more oxygen because the graph is lower here, and that's exactly Bohr's effect. What Bohr's effect is. So we are not asked about Bohr's effect right now. So they are saying that they are asking about the PO2 in the alveolar is 13.0 kilopascals and POCO2 is 5.3 kilopascal. So if PCO2 is 5.3 kilopascal, which graph of these will you look? Obviously, you look for PCO2 5.3 kilopascal. This is the graph that's relevant for you. Using figure 5.2 suggests that percentage saturation hemoglobin blood entering the pulmonary veins. So pulmonary veins will take the PO2 30.0. So we'll go to the PO2. So we know that all the PO2 is mentioned on the x-axis. This is the value 13. So with 13, we'll just drag the value up. We'll touch the graph, which is at the top, and we'll try to read off the value here. So this is 98%. Our answer will be 98%. So if they had asked us for 10.7, would have touched the lower graph and would have read it from here, that probably would have been 96%. Use the information figure 4.2 to state and explain the effect of increasing PSUT in muscle and the strength during strenuous exercise. Just need to explain Bohr's effect, that how in Bohr's effect you have more hemoglobinic acid forming and more O2 gets delivered. So increasing PSO2 in the muscle during strenuous exercise increase the rate of CO2 entering into the blood. When more CO2 enters, there's production of H plus ions in the red blood cell. H plus bind to hemoglobin forming HHV. The formation of HV causes hemoglobin to lose more oxygen to the muscles. This effect of PCO2 on oxygen delivery is called Bohr's effect. Figure 4.2 shows the effect of PCO2. 10.9 kilopascal tissue would get 38% of oxygen from the blood if its PO2 is 6 kilopascal, while the tissue with PCO2 of 5.6 kilopascal would only get 26% of oxygen from the oxygen blood. What have I done? I've just taken one value and then just extrapolated it for these. So you can just do this yourself. So for example, if this is the value of 3 kilopascals, and if I try to just take it up at the lower graph and I to read off the value here, so the tissue which is having a high PCO2 will end up receiving this much of oxygen from the blood. Whereas 60% of hemoglobin will stay saturated with blood, will stay saturated with oxygen, and it will only allow 40% of oxygen to be lost. Similarly, if I take, again, if I take same value of 3 kilopascals, but the tissue is the one which is having a low PCO2, now in this case, this tissue is actually allowing 74% of red blood cell to stay saturated with oxygen, whereas only 26% of oxygen is being delivered to the tissue. So obviously, which tissue is receiving more oxygen? The tissue which is having a high PCO2, because here there will be a high HHB formation. I hope that was helpful for you. That's it from my side. Thank you so much.